Now we will talk about the vector DNA, right? And as I already said that the vector that we will consider here is the PET21. And before I enter into the cloning, I want to tell you, along with I will try to explain you what are the different terms, how how we choose a particular thing, why we take that particular thing, and what are the properties and etc. So I will help you to build a complete background, a complete base to understand any kind of molecular cloning technique or method. So now we will talk about the PET21 DNA, PET21 vector, okay? And for example, if you are new and you don't know anything, how to find a vector and how anything beyond the name PET21, how you will do. So we will go to Google and we will type here the vector map of the PET21 DNA, and then you got these couple of links, okay? So I used to like this AdGene. AdGene is a place where you can get um, the information of all the available plasmids, whether they are expression vector, bacterial, eukaryotic, insect cell line. So this is a repository, you can say, where you can find the information about all the vectors, okay? So what I open this link here, and then I will show you what you have to keep in mind when you want to understand about a particular vector, right? So here you can see it's just name PET21. Which kind of plasmid it is. It is a bacterial expression. It's not only for the cloning, but it's especially designed for the expression, right? I will tell you what are the properties of expression vector just shortly after this. So expression level is high, so this is a high expression level vector. We should know the size. There should as a size limit. So I think the vector should not be beyond 6 kilodalton or something, because you will also incorporate some 1 kilodalton or 2 kilodalton of your gene sequence. So roughly 6 kilodalton is a good size here. Then these are the sequencing primer. You need the sequencing primer once you perform the cloning. So once your cloning is done, once you inserted the desired gene into the vector DNA, you have to confirm whether the gene is actually placed at the right place or not inside the gene, right? So for this you have to do sequencing. I will explain you about the little bit about the sequencing thing, how you do this, and what are these, how these vector functions, okay? This vector also got some protein tags for for purification or for detection. So it got the T7 tag, a T7 protein tag, a T7 small protein tag at the N terminus, and it got a his tag, his 6. So his tag is a 6 histidine in consecutive sequence. So his. So the 6 histidine. So the histidine tag is at the C terminus, right? And this particular plasmid has a resistance of ampicillin. So if a bacterial cell has this plasmid, so because of this plasmid, this gene, the ampicillin resistance gene, which is carried by this plasmid, when you will insert this plasmid, when you insert the PET21 in a bacterial cell, upon expression, this plasmid will confirm ampicillin resistance. So for example, you are transforming a lot of bacteria with PET21 vector, and you grow that transformants on a plate, on an agar plate, which contains the ampicillin, okay? So it's obvious only those bacterial cell will grow which has this vector, because this is the vector which will confer the ampicillin resistance. So those bacterial cells which will not transform by this vector, they will die because they cannot survive in an antibiotic, which is ampicillin here. So this ampicillin, this resistance is also very important for us, which antibiotic resistance is conferring. And ampicillin and kanamycin are very common to antibiotic resistance, which you will find in all the bacterial vector, right? No, this is not important for us here. Yeah, okay, so only these are the tags which we needed, okay? So I would say again, just to recall you, here you can again find the name, which is the plasmid type and expression level. These two you need. You have to look for the size, what size it is, You also need the sequence of sequencing primer for this vector. You should know what kind of tag it automatically has. I mean, you can also insert some tag externally, right, using the cloning method, but just to sometimes it's very important. And it got which kind of antibiotic resistance it has. So these are information that you can get here. The next I will explain you a little bit about this vector, what these sequences are here. So let's make it a little bit big. You can see here some very common features. So from the theory which you must have studied during your college days is that a vector is a DNA, a plasmid or a vector. So we call the plasmid to a vector, and a vector can be a plasmid. Okay, so all the plasmid or vector should have a origin of replication, so they can replicate independently. They can replicate independently inside the bacterial cell. Okay, so here in this case it got a PBR322 origin of replication, so it's stretched from roughly, so these are the sequences here. Okay, so if it is 5443 base pair, the origin of replication is roughly extending from 3200 to something 3900 or something, right? 800 something. So you can have an idea here. So this is origin of replication. As I said, it should have second feature is that it should have antibiotic resistance gene. So you can see it's got the ampicillin resistance, this red thing here. And it also got the LAC-I, which is the LAC repressor protein gene, so it will also encode for a LAC-I. And it also has a very small fragment of tetracycline, but it's not important for us here. It's not important for us. And then this is ampicillin promoter. So this is ampicillin resistance gene with the promoter. The promoter is required for any transcription. So when this plasmid will be inserted in a bacterial cell, with the help of this promoter, the ampicillin gene will express, and then it will confer the antibiotic resistance. Then the important section is here, this one. We call this multiple cloning site. And there are several other features in the multiple cloning site here, which I will explain you. So let's recall from our basics. If we want to transcribe any gene, we need a promoter, we need a gene sequence, and we need a terminator. So it's roughly it would be like this. So you can say that if this is your gene segment, it should roughly have a promoter here. This is a promoter region, this may be your gene, and then somewhere here is a terminator region. So this is promoter, this is your gene segment, and this is terminator. So if you want to insert a gene and you want to express that gene, you should have two features. What's a promoter, and one is terminator sequence. So these are the rules for transcription. Okay, if you want to translate that gene, I mean after transcription, if you want to express or you want to translate that gene into a protein, so you want to translate an mRNA into a protein, you should have an additional feature which we call ribosome binding site. So this, I hope that you remember from your textbooks, that the Kozak sequences in bacterial expression. So the Kozak sequences are roughly they offer not the Kozak sequences, I'm really sorry, it's a signed Dalgarno sequence, which is present in next to the promoter region, so just before the AUG. So you know that the AUG is the start codon. So if the start codon is here, let me just draw this. If this is the start codon, if this is a start codon, and before that there is a sequence, a stretch called Dalgarno sequences, or you can say it's simply ribosome binding site. So for for transcription, you need a promoter. So the RNA polymerase will bind to this promoter region and then transcribe this gene into an mRNA, and it needs a terminator so that once the gene is finished, after recognizing the terminator sequence, the RNA polymerase can just leave off so that it can only synthesize the gene that you need. Okay, so only this much. And if you want to translate the sequence, then next you need a ribosome binding site inside the gene. So there is a ribosome binding site where the ribosome can recognize the sequence and bind and can start the translation. Okay, so I hope these 2 points are very clear to you. So here you can see this, there is a T7 terminator here and there is a T7 promoter here. Okay, so now what we can see here, after T7 promoter there is a lac operator. There are some restriction enzymes, XHO1, NOT1, EAG1, HIND3, SAL1, SAC1, etc. So what are the features that you again should keep in mind? Okay, so you should know whether, T7 terminator, you should know whether it has a promoter, so it got T7 promoter and T7 terminator. Great. The next we need to know whether it got several restriction enzymes. Yes, it got series of restriction enzyme, and you can insert your gene in any of these combination. While you are doing this, you just have recognized here that it also got a ribosome binding site. So RBS means is a ribosome binding site. So it's got a T7 translation entry, it's a ribosome binding site. Okay, now here is something you should know. So these are different genes. You wanted to preserve your ribosome binding site, okay, and then you want to insert your gene, because after the transcription, which will be initiated by this promoter, you also want to do the translation because you want to express your protein, right? So you need the ribosome binding site. So if you want to insert your gene in this multiple clonic site, you should use from NDE1 to XHO1. So you can use any of these restriction enzyme combinations. Okay, this is important here. There is another question that you may ask me, because you see a lot of restriction enzyme here. I mean, you can ask me why there is a PST1 restriction enzyme here, can't you clone a gene here? There are a lot more enzymes. That EcoRV restriction enzyme site, cutting site is here, that HPA1 cutting site is here, and etc. So you can ask me, can you insert a gene here or not? So I will answer that you cannot insert a gene here. You can see these restriction enzyme cutting site has certain property. What is that property? Maybe, for example, let's concentrate here. So the ampicillin resistance gene has a PST1 restriction enzyme cutting site, right? But you cannot insert a gene here. Why? As you can see, if you will cut this part here, as this is a PST1 site, if you cut this part here, you will disrupt the ampicillin gene, right? Because you will cut and then this part will go this side, this one goes this side, and you will insert a gene. So then you are disrupting the ampicillin resistance gene. Once you are disrupting the ampicillin resistance gene, you cannot use this because you cannot select it. There is no antibiotic resistance, right? So you want to preserve it. You cannot insert a gene. But then you can ask me why there is a restriction enzyme site. For example, for some reason you want to convert this ampicillin resistance gene to, I mean, you do not need the ampicillin resistance, you need some other antibiotic resistance. For example, kanamycin, you need kanamycin antibiotic resistance here. So what you can do, you can cut this vector with PST1, then this will cut it here, the ampicillin resistance will disrupt, and you can insert a kanamycin resistance gene here, or you can just insert any sequence here so that you disrupt the whole ampicillin resistance cassette or whole ampicillin resistance gene, and then no longer it is in use, and then at any other place, let's say maybe here, you can put kanamycin resistance gene. So these features have certain importance, which is not required for you at the very beginning, but just to let you know that though these are also restriction enzyme, but you cannot use them for the cloning. You can only use for the cloning because this is the segment which is giving you a T7 promoter, which is giving you a terminator, which is giving you a ribosome binding site. So this is the perfect thing where you can put your gene. So this is a general information for a vector for you. I will also explain you something here. So these are here. So the BLA coding sequence means for the antibiotic. It got the origin, again it's got like I encoding gene, terminator, there's a his tag, it got multiple cloning site, it's from BAMH1 to XHO1, BAMH1 to XHO1, so that means you should insert your gene somewhere here. I mean, you can also use this part without a problem, okay, so don't worry. And etc. And then these are the sequence which they have amplified for you in the sequence basis and tell where these are cutting sites. So you can see, so there is a BGL2 restriction engine cutting site is here before the T7 promoter, and then there is a lac operator. You know, these expressions are induced by the IPTG, isopropyl sorry, IPTG, yes, isopropyl thiogalactopyranoside. So I mean, this we will tell you about how this expression thing, but just for a minute. So you can see here there is a BGL2 and XBA1 cutting site here, which is flanking the T7 promoter and lac operator. So if you want to remove this segment here, if you just want to remove this segment, you just simply cut this with BGL2 and XBA1 and you can just get rid of, but you do not want it, right? And then you can see here is this ribosome binding site here, see, and then there is NDE1, NHE1, these different tags, then there's this T7 tag. So if you need the T7 tag, you should not cut with these two enzymes, but you should cut between BAMH1 to XHO1, okay? And then there's a his tag and terminal. So now I will tell you what are the places where you can put your gene, where you can clone your gene, okay? So T7 tag is not always necessary because we generally not prefer N-terminal tag in the bacterial system. We generally prefer a C-terminal tag because N-terminal tag can affect the folding of protein when you express it, okay? So generally we do not prefer. And so if we do not want a T7 tag, our multiple cloning site is extended now from NDE1 to XHO1. So we can place a gene between NDE1 to XHO1 here, and then we will have the his tag. And then you can see here at the very end here there's a T7 terminator, okay? So these are the very important properties for you. Then there is one more thing which I should tell you about how these sequencing primer works here. So for example, you inserted a gene between NDE1 and HindE3, okay, and then you wanted to know whether your gene is inserted properly or not. So what you will do here, you will take this primer sequence, which is from TAA to AGG. You will take this primer sequence, this primer from this sequence to this sequence, and you will take the T7 terminator primer from CGA to GCC. So you will take these two primers and you will perform a sequencing analysis. The sequencing will amplify your gene from this region to this region. So whatever will be inserted between these two regions you will get in a sequences. So you perform DNA sequencing, okay? And then you can analyze by nucleotides, just reading all the nucleotides, whether your gene is inserted properly or not, okay? So this is the information that I wanted to tell you about a vector, and I hope you actually understood this about these different vectors. Just to quickly to complete this, I will I will just print this structure for you, so that you can also get the feeling when you wanted to look at it. Just in 1 second. Yes, I know it's very technical here, but I want you to have a proper feeling so that you can understand everything properly. So what is the place where you can get all the information is at Gene. You just type this thing in Google and you will find it. That's it. Great. Thank you.